Hello everyone, a very welcome to VYAS Edification Quota. So today in the series of NCERT discussion, we are going to discuss NCERT questions of a chapter Microbes in Human Welfare from class 12. The first question of this chapter is that bacteria cannot be seen with the naked eyes, but these can be seen with the help of a microscope. If you have to carry a sample from your home to your biology laboratory to demonstrate the presence of microbes with the help of a microscope, which sample would you carry and why? Of course, you will carry curd. Curd is the best sample by which you can test the presence of microbes in it. Why? Because curd contains numerous of lactic acid bacteria or lab or lactobacillus. These bacteria produces acid in the milk which coagulates and digests the milk proteins. So this can be the best sample to test the presence of a microscope. Even a drop of curd can contain millions of lactobacillus or lactic acid bacteria in it. So this is the answer. Now let's write the answer of this question. So answer number 1, curd can be used as the sample to test the presence of lactic acid bacteria or lab or lactobacillus or lactobacillus. These bacteria produces acid which coagulates the milk, which coagulates and digests the milk proteins, which coagulates and digests the milk proteins, Even a drop of curd contains millions of lactobacillus. So this is the complete solution of question number 1. The next question we have is, give examples to prove that microbes releases gases during metabolism. Alright, so we have to give some examples to prove that during metabolism of microbes, they produces gases. So the first example of this is the puffed up appearance of dough of idli and dosa. During this, the reason behind this is carbon dioxide gas is released during the process of fermentation done by bacteria in them. The next example or the another example of this can be the treatment of wastewater. During the treatment of wastewater, when the sludge is being digested by the bacteria, these bacteria produces a mixture of gases which involves carbon dioxide, hydrogen sulfide and methane. So all these are the examples of metabolism of bacteria which produces gases. So let's write answer. Answer number 2. The examples to prove that microbes releases gases during metabolism, during their metabolism. Are, the first example is puffed up appearance. The puffed up appearance of dough, of dosa or idli, because this is because carbon dioxide released or releases by bacteria during the fermentation of dough, during the fermentation of dough. The next example for this is during the digestion of sludge, during the digestion of sludge at the time of wastewater treatment, at the time of wastewater treatment, bacterias produces a mixture of gases, such as methane, carbon dioxide and hydrogen sulfide, H2S, hydrogen sulfide. Clear? So these are the examples and this is the written solution, complete solution of question number 2. Now the next question, question number 3, we have to discuss is in which food would you find lactic acid bacteria and mention some of their useful applications. So you will find lactic acid bacteria in milk and its product. You will find lactic acid bacteria or lactobacillus in milk and its products, in which it produces lactic acid. These lactic acid bacteria or lactobacillus produces lactic acid during the fermentation of carbohydrates. Right? Now if I talk about the application of these lactobacillus or lactic acid bacteria, the first application of these is they are found in milk, right? They produces lactic acid in milk, which coagulates and partially digests the proteins of milk. It also adds vitamin B12 in the milk, which enhances the nutritional quality of a milk. The second application, the second useful application of these lactic acid bacteria is they are found, they play a very beneficial role in our stomach in checking disease causing microbes. So these are the applications and you will find these lactic acid bacteria or lactobacillus in milk and their products. Now let's write the answer of this question. Question number 3, answer number 3, we can find these lactic acid bacteria we can find these lactic acid bacteria in milk, and its products, in which they produces lactic acid which coagulates and partially digests the milk. Actually, this is the application. You can find these lactic acid bacteria in milk and its products. Then we have to mention some of their useful applications. So this is application. The milk, they are found in milk, in which they produces lactic acid which coagulates and partially digests the proteins of milk. They also adds vitamin B12 and enhances the nutritional quality of milk. The second application is these bacteria plays a very important role in checking the disease causing microbes in our stomach. That is why we are advised to eat curd, because it contains so many lactobacillus or lactic acid bacteria which plays a very important role in our stomach in checking disease causing microbes. So these are the useful applications of these lactic acid bacteria, and you will find these lactic acid bacteria in milk or its products. So this is the complete solution of question number 3. Now let's move on to the next question, that is question number 4. In this question we have to name some traditional Indian foods made up of wheat, rice and Bengal gram or their products, which involves the use of microbes. Alright, so to make Indian traditional dishes made up of wheat, rice and Bengal, we have some examples for that, such as idli. To make idli or dosa, we ferment the rice powder with the help of bacteria. To make dosa or idli, rice powder is fermented with the help of bacteria. And to make breads, which is made from wheat, which is made with the help of wheat, this wheat is fermented with the help of a yeast, particular yeast which is known as Saccharomyces cerevisiae, and thus breads are made, right? Then if we talk about the next, that is your Bengal gram. So to make gatta, which is made up of black gram, it is also fermented with the help of microbes. So these are some examples of traditional dishes which is made up of wheat, rice and Bengal gram, in which the microbes are also involved. Now let's write answer of this question. Answer number 4. So to make dosa and idli, rice powder is fermented. Rice powder is fermented with the help of bacteria, and to make bread, which is made by wheat, is fermented by yeast, that is Saccharomyces cerevisiae, and to make gatta, made up of black gram, is also fermented by bacteria. Also, bacteria or microbes, any microbes are used to ferment the dishes like fishes and bamboo shoots, etc. So this is a complete solution of question number 4. Now let's discuss question number 5. In which way have microbes played a major role in controlling diseases caused by harmful bacteria? So first of all, what is antibiotic? Antibiotics are the substances which are produced by the natural metabolic processes of bacteria and fungi as their self-defense against other microbes. So antibiotics are the substances which are produced by the natural metabolic processes of bacteria and fungi as their self-defense but against other microbes. Among bacteria and fungi, most of the antibiotics are produced from bacterial genera. The highest number of antibiotics which are produced from the bacteria are from genera Streptomyces and Bacillus. And if I talk about the fungi, the highest number of antibiotics which are produced from the fungi are from genera Penicillium and Cephalosporium. So this is the answer. Now let's write the answer of this question. Answer number 5. Antibiotics. We will start with antibiotics. Antibiotics are the substances produced by the natural metabolic process in bacteria and fungi as their self-defense but against other microbes. Now, among bacteria and fungi, the most antibiotics are produced by bacteria, and the highest number of antibiotics produced from bacteria from genera Streptomyces and Bacillus, Streptomyces and Bacillus. Now the highest number of antibiotics, the highest number of antibiotics are produced from fungal genera are penicillium and cephalosporium. So this is the complete solution of question number 5. Now let's discuss question number 6. Name any 2 species of fungus which are used in the production of antibiotics. So antibiotics are the chemical substances, we know antibiotics are the chemical substances which are produced by the microorganisms like bacteria and fungi to treat and prevent the diseases caused by other microbes such as bacteria and parasites. Clear? Now if we talk about the 2 common species of fungus which are used to produce antibiotics, such as Penicillium notatum. Penicillium notatum is the common fungus which is used to prepare the antibiotic penicillin. The another species of fungus which is used to produce antibiotic is Cephalosporum acremonium, which is used to produce the antibiotic known as cephalosporin. So these are the species of fungus which are commonly used to produce antibiotics, and I hope you know that what is antibiotic. So let's write the answer of question number 6. Antibiotics are the chemical substances. This is C, chemical substances which are produced by the microorganisms like bacteria and fungi, So antibiotics are the chemical substances which are produced by the microorganisms like bacteria and fungi to treat and prevent the organism by the microbes, by the other disease causing microbes. These antibiotics may either kill these microbes or inhibits the growth of them. Now the most common species of fungus used to prepare antibiotics are number 1 Penicillium notatum, Penicillium notatum, which is the source of penicillin, which is an antibiotic, penicillin. And the next species is Cephalosporium acromium, which is the source of an antibiotic known as cephalosporin. Cephalosporin. So these are the antibiotics. Penicillin and cephalosporin are the antibiotics which are prepared from the common species of fungus such as Penicillium notatum. Penicillin is produced from the Penicillium notatum and cephalosporin is prepared or produced from the species of a fungus such as Cephalosporium acromium. So this is the complete solution of question number 6. So now here we have next question, question number 7. What is sewage and in which way can sewage be harmful to us? So what is sewage first of all? Sewage is a municipal waste water. Sewage is a municipal waste water collected from cities and town homes which contains toilet, kitchen and bathroom waste. Now these waste water contains large amount of organic matters and many pathogenic bacteria which are very harmful to humans as they can cause some diseases, many diseases such as typhoid, cholera, etc. So this is sewage. And in which way can sewage be harmful? Because it can cause several diseases to human beings because it contains many organic matter and many pathogenic bacteria or microbes in it. So let's write the answer of question number 7. First of all, we'll write what is sewage. So sewage is the municipal waste water collected from cities and townhomes which contains toilet, bathroom and kitchen waste wastes. Now these waste water contains organic matter, large amount of organic matter, and many pathogenic microbes which are very harmful, to humans. As they can cause several diseases such as typhoid, jaundice, etc. Clear? So what is sewage? Sewage is the municipal wastewater which is collected from the cities and town homes, which collects many pathogenic microbes and a large amount of organic waste, organic matter, which is harmful to the human beings, as they can cause the diseases. So this is the complete answer or solution of question number 7. Now let's discuss next question, that is question number 8. What is the key difference between primary and secondary sewage treatment? So we have to talk about the differences between primary and secondary sewage treatment. What is primary sewage treatment? First of all, the primary sewage treatment is the mechanical process which involves the removal of solid particles or solid waste from the sewage. Whereas what is secondary sewage treatment? Secondary sewage treatment is the biological process which involves the digestion of organic waste present in a sewage by bacteria. Right, so this is the key difference between primary sewage treatment and secondary sewage treatment. Next difference between these two is the primary sewage treatment. The primary sewage treatment is inexpensive and less complicated process, whereas the secondary sewage treatment is very expensive process as well as it is more complicated. So these two are the key differences, basic differences between primary and secondary sewage treatment. Now let's write the answer, and we will write this answer in the tabular form by making a column. In the left column we will be writing primary treatment, primary sewage treatment, and in the right column we will be writing the secondary sewage treatment. So here primary sewage treatment, and in the right column secondary sewage treatment. Clear? Now let's write the difference between, the first difference between these two, the primary sewage treatment. The primary sewage treatment is the mechanical process. First of all, it is the mechanical process which involves the removal of solid waste from sewage. Now about secondary sewage treatment, the secondary sewage treatment is a biological process as it involves bacteria which digest the organic waste present in sewage. The next difference between these two is about primary sewage treatment. It is inexpensive. It is inexpensive and less complicated process, whereas this one, the secondary sewage treatment, is very expensive and a more complicated process of sewage treatment. So this is the complete solution of question number 8. The next question we have is 9th question. Do you think microbes can also be used as source of energy? If yes, then how? So microbes produces various or different types of gaseous and products during their growth and their metabolism. Yes, microbes do produce various gaseous and products during their growth and metabolism, which we can use as a source of energy. For example, you must have heard about biogas. Biogas is a mixture of gases which is produced by the microbial activity and can be used as a fuel. Another example is alcohol. Alcohol is also can be used, or first of all it is mixed with other fuels to obtain it as a source of energy. So these are the examples, and this is how bacteria can help, or bacteria can produce some substances which can be used as the source of energy. So let's write answer of this question number 9. Answer 9. Bacterias or microbes, bacteria or I may write microbes only. Microbes produces various kind of gaseous and products during their growth, and metabolism. Now, microbes produce various kinds of gaseous and products during their growth and metabolism, which can be used as the source of energy, such as biogas. Biogas is a mixture of gases, which is produced by the microbial activity and is used as fuel. Another example, another product such as alcohol can be mixed with other fuels and used as fuel, right? So this is how we can use the substances which is produced from the microbes, and we can use them as the source of energy. So this is the complete solution of question number 9. Now let's discuss question number 10. Microbes can be used to decrease the use of chemical fertilizers and pesticides. Explain how this can be accomplished. Alright, so we can use various microbes as biofertilizers, and they enrich the nutrient quality of a soil. Some microbes such as bacteria, fungi, cyanobacteria can be used as biofertilizers instead of using chemical fertilizers. What they are doing, what these microbes are actually doing, they are enriching the nutrient quality, nutrient content in the soil. Hence, they are increasing the fertility of a soil. So we can use them as fertilizers instead of using any chemical fertilizer. Now the examples of these microbes are rhizobium. First of all, rhizobium is a bacteria which is found in the roots of a leguminous plants or higher plants, right? So rhizobium is a bacteria which forms nodule. What it does actually, it forms nodules in the roots of a leguminous plant, and it is found in the symbiotic association with the roots of a leguminous plant. What it does actually, it fixes the atmospheric nitrogen, it converts the atmospheric nitrogen into its organic form so that plant can use it easily. Because plants are not capable of using or obtaining the nitrogen in its atmospheric form or in its original form which is found in the atmosphere. So rhizobium is a bacteria which forms nodules in the roots of a leguminous plant in symbiotic association with them, and it fixes the atmospheric nitrogen. It is converting the atmospheric nitrogen into its organic form which is used by the plants. Now, second example is also from bacterial category, that is Azotobacter and Azospirillum. Azotobacter and Azospirillum are also the examples of bacteria which are not found in the symbiotic association with other organisms. They are freely living organisms. They are freely living bacteria. The function of these two bacteria, Azospirillum and Azotobacter, is actually the same with rhizobium. That means Azotobacter and Azospirillum are also fixing the nitrogen into another form so that plants can use it. But Azotobacter and Azospirillum are the free living bacteria, whereas the rhizobium is found in symbiotic association with the higher plants. The next example is from category fungi, because we can use the fungi as biofertilizers. Now, many members of the genus Glomus, which is a fungi, many members of the genus Glomus, which is a fungi, it actually converts nitrogen, atmospheric nitrogen, into different form, and it is found, many members of the species of Glomus found in the symbiotic association with the plants, which is called as mycorrhizae. Now, in that symbiotic association, what fungi is doing? Fungi is actually obtaining the phosphorus. Fungi is obtaining the phosphorus from soil and it is giving it to the plant. Fungi in the symbiotic association called as mycorrhiza, it is obtaining the phosphorus from soil and it is giving it to the plant. This fungi in the mycorrhiza also increases the tolerance level of plant to salinity and drought, so that overall increase in development and growth of a plant can be seen. Also, this fungi is developing resistancy to the pathogen, root pathogen to the plant. So this is the working of fungi in the symbiotic association called as mycorrhizae. The next category is of cyanobacteria. Now, we have so many organisms, we have so many bacteria in cyanobacteria category, that is, for example, Anabina, we have Nostoc and Oscillatoria. These three organisms, Oscillatoria, Nostoc and Arabina, are from category cyanobacteria. They are also fixing the atmospheric nitrogen into aquatic forms of plant and as well as terrestrial forms of plant, right? So this is how these all microbes from cyanobacteria, fungi and bacteria can be used as biofertilizers instead of using chemical fertilizers. Now let's write the answer of this question here. Answer number 10. So microbes, we'll start like microbes can be used as biofertilizers microbes can be used as biofertilizers instead of chemical fertilizers. Chemical fertilizers. The microbes, like what kind of microbes or which microbes can be used as biofertilizers? That is bacteria, cyanobacteria and fungi. So the microbes like bacteria, fungi and cyanobacteria. Cyanobacteria can be used as biofertilizers and they enrich the nutrient quality, nutrient content actually in the soil. Right? So the microbes like cyanobacteria, bacteria and fungi can be used as biofertilizers instead of using chemical fertilizers. They enrich the nutrient content into the soil, hence increasing the fertility of a soil. Then let's talk about the examples. So examples are as follows. The first one we have is rhizobium. Rhizobium. This bacteria forms nodules in the roots of leguminous plants. In the roots of leguminous plants in symbiotic association fixes atmospheric nitrogen into its organic forms, which can be used by plants. Clear? The next example is Azotobacter and Azospirillum, which are also the examples of bacteria only. They are bacteria. So Azospirillum and Azotobacter are freely living bacterias which also fixes the atmospheric nitrogen. Clear? So these are the examples of these are the examples of bacteria which fixes the atmospheric nitrogen so that the plants can use it easily. Then the next example is from fungi. So many members from the genus Glomus, which is a fungi, also converts, I am writing in continuation on the next slide, also fixes the nutrients thus enriching the nutrient quality of soil. What it does? First of all, it is found in the symbiotic association. The many members of fungi found in the symbiotic association with plants. So this fungi, the fungi or the members of Glomus found in the symbiotic association with plants forming mycorrhiza. In which this fungi obtains or absorbs phosphorus from the soil and give it to the plant. So this fungi absorbs phosphorus from the soil and give it to plant. The next thing, this fungi in the symbiotic association mycorrhiza, what it does? It increases the tolerance level. It increases the tolerance of plant to salinity and drought, so that plant can grow easily. And the next thing, it develops resistancy. It develops resistancy to the root pathogens. Right. The next example is from cyanobacteria. Cyanobacteria such as Anabina, nostoc and Oscillatoria. These three cyanobacteria such as Anabina, nostoc and Oscillatoria, they also fixes the atmospheric nitrogen in aquatic and as well as terrestrial forms. Clear? So this is the answer. This is the complete solution of question number 10, in which we have already talked about the microbes which can be used as biofertilizers instead of using chemical fertilizers. So this is the complete solution of question number 10. Now we will be discussing question number 11. 3 water samples, namely river water, untreated sewage water and secondary effluent discharged from a sewage treatment plant were subjected to BOD test. Then the samples were labeled A, B and C, but the laboratory attendant did not note which was which. The BOD value of the 3 samples A, B, C were recorded as 20 milligram per liter, 8 milligram per liter and 400 milligram per liter respectively. Which sample of the water is most polluted? Can you assign the correct label to each, assuming the river water is relatively clean? So if we talk about, we have given BOD in this and we need to perform BOD test. The laboratory attendant was actually performing a BOD test. What is BOD? BOD is a biological oxygen demand, which directly relates to the pollution or organic matter present in the water. So BOD is the direct measure of organic matter present in any water. More the BOD is, more the biological oxygen demand is, the more the polluted water is. So according to the knowledge of BOD or biological oxygen demand, we can assign the BOD values to the given samples. So let's write the answer of 11th. So first of all, we'll write biological oxygen demand. Biological oxygen demand, that means BOD, is the direct measure, of organic matter present in water, right? Now according to the knowledge of BOD, we can assign these values to the samples, to the given samples. So first we can assign this one, the 20 ml per liter. 20 ml per liter is a BOD. We can give it to the untreated, we can give it to the secondary effluent which is discharged from the sewage treatment. See, we know that more the BOD is, more the water is polluted. So first we'll start with the highest value of BOD. Let's start with the highest value of BOD. And what is the highest value of BOD among these 3 is 400 milligram per liter. So 400 milligram per liter is given to, is it given to river water? Is it given to untreated sewage water, or it is given to secondary effluent discharge from sewage treatment? Of course, it is untreated sewage, because the BOD level is so high. And if the BOD level is so high, that means there will be so much of organic matter present in that effluent. So here we can see that 400 milligram per liter is given to untreated sewage water. Then let's come towards the next one, that is 20. Lesser than 400 we have 2 values, that is 20 and 8. So we'll talk about 20. 20 ml per liter it can given to the secondary effluent discharged from a sewage treatment plant. So this is given to secondary effluent discharged from sewage treatment. And of course, the river water will have less BOD as it does not contain any organic matter or less organic matter comparatively. So the last value, which is the lowest value of BOD, that is 8 milligram per liter, is the value we can give to the sample river water. River water, as it contains less amount of organic matter in it. So now here we can write a note that the more is the BOD is more, the BOD is more, the water is polluted. More the water is polluted. So you can clearly identify which value of BOD can be given to which kind of sample. So this is the complete solution of question number 11. Now let's discuss next question, question number 12. Find out the name of the microbes from which cyclosporin A, which is an immunosuppressive agent or immunosuppressive drug, and statins, which is a blood cholesterol lowering agent, are obtained. So these two drugs are obtained from the microbes, and these two microbes such as cyclosporin A is obtained from Trichoderma polysporum, and the next one, that is statin, is obtained from Monascus purpureus. So these two are the organisms. Let's write answer number 12. The cyclosporin A the cyclosporin A, which is an immunosuppressive drug, is obtained from Trichoderma polysporum. And the next, the statin, which is a blood cholesterol lowering agent, is obtained from Monascus purpureus. These are the two organisms from which the cyclosporin A and statins are obtained. So this is the complete solution of question number 12. Now let's discuss next question, that is question number 13. Find out the role of microbes in the following and discuss it with your teacher, that is single cell protein and soil. So first we will be discussing single cell protein. What is single cell protein? Single cell protein, or SPR proteins, is a type of protein which is obtained from either from pure or from mixed cultures of bacteria, cyanobacteria, etc. that have 30 to 70% of protein content in their dry mass. For example, Methylophilus and Methylotrophus, these are the bacterial species which produces proteins, and then they can be used by humans and other animals. So this is your single protein species of bacteria. Then next we have is example of cyanobacteria, that is chlorella. You must have heard about chlorella. Chlorella is first of all a single-celled green algae belonging to the class Chlorophyta, which produces proteins. It have essential amino acids needed for tissue building and maintenance of the metabolic processes. So it is used as a source of protein. So this is single cell protein or SCP. Then if we talk about soil. So the microbes like protozoans, bacteria, fungi, etc. in the soil, they break down the organic matter into soil. They also act as decomposers and a part of detritus food chain. This detritus food chain helps in the replenishment of soil and hence increasing the nutrient of a soil. So this is about question number 13, and let's write the answer of this question. First we'll be writing about SCP or single cell protein. So single cell protein or SCP is a protein which is obtained from pure or mixed culture of bacteria, cyanobacteria, etc. For example, methylophilus and methylotrophus. Produces proteins which is used by humans and animals. The next example for SCP or single cell protein is chlorella. The next example is chlorella. A single-celled green algae belonging from class Chlorophyta. Serves protein and contains essential amino acids needed for tissue building and maintenance of metabolic processes. So this is SCP. Now let's write about soil. The microbes such as protozoans, bacteria and fungi in soil break down the organic matter. break down the organic matter. They also act as decomposers and part of detritus food chain. This food chain helps in soil replenishment, and increases its fertility. So this is what about SCP and soil. Now this is the complete solution of question number 13. Now let's discuss the second last question of this chapter. Arrange the following in the decreasing order, most important first of their importance for the welfare of human society. Give reasons for your answer, and we have to arrange all these: biogas, citric acid, penicillin and curd. So according to our knowledge, we will give it as the first position. Then we'll talk about biogas, curd and citric acid. If I talk about the most important form with its importance, so penicillin is what? Penicillin is an antibiotic. It is an antibiotic which is obtained from the Penicillium notatum, and because it is an antibiotic, it is used to cure many bacterial diseases. Then the next one we have is biogas. Biogas is an eco-friendly source of energy, particularly in rural areas, so it is of importance. And then we have curd. Curd is an digestible. Curd is very good, and it is containing digestible vitamins, and it can be digested by humans easily. Then we have citric acid. Citric acid is mainly used as preservatives in many food items. So this is the arrangement of all these items, of all these things which we have given. Then let's write answer 14. The first one we will write about penicillin. So penicillin, it is an antibiotic. First of all, it is an antibiotic which is obtained from Penicillium notatum and is used to cure many bacterial diseases. The next we have is biogas. So next, first position is given to penicillin. Next is biogas. It is an eco-friendly source of energy, particularly in rural areas. Then third is given to the curd. It is easily digestible vitamin rich food. Curd increases vitamin B12. Curd is rich in vitamin B12, which is easily digestible by us. Then the last is citric acid. It is mainly used for the purpose of preservative in many food items, So this is the answer. This is the complete solution of this question number 14. Now let's discuss the last question of this chapter. How do fertilizers enrich the fertility of the soil? So first of all, the biofertilizers are a type of fertilizers which are obtained from the living microorganisms such as bacteria, cyanobacteria, etc. When these biofertilizers such as bacteria, cyanobacteria are applied to the field, they increases the availability of nutrients used by the plants. They increases the availability of nutrients used by the plants into soil. They also increases the soil fertility, and many species of cyanobacteria and bacteria are able to fix the atmospheric nitrogen into other forms so that plants can use this nitrogen, because plants are not capable of using nitrogen in its atmospheric form. Then if we talk about the advantage of biofertilizers, they are eco-friendly and they are cost effective. So this is the advantage of biofertilizers. Now let's write the answer of this last question, question number 15. So answer 15 is biofertilizers are the fertilizers obtained from living microorganisms. When these biofertilizers are applied to the field, when these biofertilizers are applied to field, they increases the availability of nutrients into soil to the host plant. Thus increasing the fertility of soil. Many species of cyanobacteria and bacteria have an ability to fix the atmospheric nitrogen If I talk about the advantages, the advantage of these biofertilizers is that they are eco-friendly and cost effective. So this is the complete solution of question number 15, and with this we have completed this whole chapter, and this is all for today. Thank you and have a nice day.